Hello everyone and welcome to this webinar. First, let me introduce myself. My name is Najah Abi-Gerges. I am VP of R&D at AnaBios here in San Diego. I have been doing drug discovery for the last 18 years and my background is in cardiac research. Let's now focus on today's topic, adult human primary cardiomyocyte. To set up the scene for this webinar, let's first define myocardial contractility. Myocardial contractility, which is essential to maintain cardiac output, represents the ability of the heart muscle to contract. The contraction is caused by a process known excitation-contraction coupling. Excitation-contraction coupling is about an electrical action potential stimulus that will release the calcium from the reticulum sarcoplasmic, initiating the contraction by sarcomere shortening. As you know, drugs can decrease or increase contractility. If decrease in contractility is significant, this may lead to heart failure, and this is the case with oncology drugs. If increase in contractility is significant, this may lead to arrhythmia and also may lead to hypoxia in patients with coronary artery disease, for example. If a drug induces inotropic effect, this may limit its utility as a novel therapy, and also it may limit its tolerated efficacy dose. Therefore, it is very important during preclinical development to assess the potential of novel drugs to induce inotropic effect. This slide summarizes for you the current safety assessment strategy that is being used by most pharmaceutical companies to assess the potential of novel drugs to induce inotropic risk. Early in drug discovery, the affinity of compound for ion channel is being tested, and also the compounds are being tested in cell and tissue-based model. Challenges with the ion channel testing is the integration of the data and the data inability to predict non-ion channel mediated liability. Challenges with the cell and tissue-based models are non-human biology, species differences, and low predictivity. When it comes to the model being used in late preclinical development, they can only be used for risk mitigation. However, they can't be used to support the chemical design of novel drugs because of their low throughput. I think all of us agree that a human cardiomyocyte model is urgently needed to consistently predict inotropic risk. To develop and validate such model, there are two types of myocytes that currently can be used: the human stem cell derived cardiomyocyte and the adult human primary cardiomyocyte. Based on published data on parameters like cell maturity, sarcomeric structure, and ion channel expression, for example, it can be concluded that the adult human primary cardiomyocyte and not the human stem cell derived cardiomyocyte exhibits the physiological phenotype. Additionally, false inotropy, false negative prediction, false arrhythmia, and false prediction of false safety margin were reported in studies using stem cell derived cardiomyocyte. Based on this, it can be concluded that the human stem cell myocyte lack mature inotropic mechanism. We at AnaBios and key opinion leaders strongly believe that a human primary cardiomyocyte model may represent the opportunity to influence chemical design during preclinical development. Here at AnaBios, we have developed and validated method and a special solution to avoid ischemic damage and reperfusion injury when we recover and when we ship the human heart to our lab in San Diego. We also developed and validated SOPs and the procedures that allows us to consistently procure and experimentally interrogate human cardiac preparation to provide consistent, reliable, and predictive cardiotoxicity data at the preclinical stages. We have also worked on over 1,000 human heart, and the high quality of the human heart we are procuring has allowed us to develop cell-based and tissue-based models. The cell-based models can be used to support projects early in drug discovery, while the tissue-based models can be used in late preclinical development. Taken together, these models, they can help to assess the potential of novel drugs to induce pro-arrhythmia and also they can help to assess their potential to modulate contractility, heart rate, blood pressure, and finally their potential to induce cardiac fibrosis. We have developed a new isolation method that provides high yield of cardiomyocyte. In this slide, you see representative images of the myocyte we obtain after isolation. You can see that the myocyte retains their rod-shaped morphology, they exhibit cross striation, and most importantly, in contrast to human stem cell-derived cardiomyocyte, they only contract and relax in response to electrical stimulation. This slide summarizes for you the feature that an adult human cardiomyocyte contractility model has to have. The model has to address both negative and positive inotropic risk. It has to have validated the predictivity of a clinical outcome, and it has to be scalable to medium high throughput for supporting drug design. We have used the non-invasive brightfield imaging to record the sarcomere shortening as a marker for contractility in adult human primary cardiomyocyte. As you know, brightfield imaging comes with low technical complexity, and most importantly, there is no need to use cytotoxic reagent. This will help to fully retain the physiology of the myocyte and their pharmacological response to drug. In this video here, you can see a myocyte contracting and relaxing in response to a field electrical stimulation at 1 Hertz pacing rate. First, we wanted to confirm the ability of adult human primary cardiomyocyte to predict negative inotropes. To do this, we tested 33 clinical well-characterized controls. 27 of them are multi-channel blockers. They mainly block potassium, sodium, and calcium channels, and we used them as positive control in this study. For negative control, we used 6 selective HERC blockers. As you know, HERC blockade does not depress myocardial contractility. Each drug was tested at multiple of the 3 Cmax. This slide shows you an experiment with Verapamil. As you know, Verapamil is a calcium channel inhibitor, and it does induce negative inotropic effect. In this video, you see when we applied Verapamil, Verapamil was able to inhibit sarcomere shortening. And here you can see that the effect, the negative inotropic effect of Verapamil is concentration dependent. This slide summarizes for you the data we obtained with the 33 drugs. Looking at the typical dose response curve, you can see that HERC blockers did not inhibit sarcomere shortening, while multi-channel blockers and class 1 antiarrhythmic drugs inhibited sarcomere shortening in a concentration-dependent manner. As you can see in the summary table with the 33 drugs, from this typical dose response curve, we can derive IC50 value, and also we can calculate a safety ratio. The IC50 and the safety ratio can be used to rank a compound, and also can be used for decision making when it comes to decide if a compound has to be stopped because it is associated with a potential to induce negative inotropy, or it can be taken forward to next R&D stage because it is free of potential to induce negative inotropy. When looking at these 33 drugs, you can see all of them have affinity to ion channels. Next, we wanted to confirm the ability of the adult human primary cardiomyocyte to predict negative inotropy with compound that they don't have ion channel as their primary target. To do this, we tested tyrosine kinase inhibitors. As you know, tyrosine kinase inhibitors provide effective treatment to cancer patients. However, tyrosine kinase inhibitors induce cardiotoxicity that it does manifest in a clinic as decrease in contractility. To validate the clinical relevance of cancer agent, we tested 9 clinical well-characterized controls. 4 of them are toxic tyrosine kinase inhibitors, 4 of them are non-toxic tyrosine kinase inhibitors, and one of them is a toxic anthracycline cancer agent, doxorubicin. We tested each drug at multiple of the Cmax, and we perfused each concentration of each drug for 5 minutes. 5 minutes here has been used because we wanted to assess the potential of the myocyte to predict any potential acute cardiotoxicity. This slide shows you an experiment we conducted with sorafenib. Sorafenib is a toxic tyrosine kinase inhibitors. You can see, if I speed up the video for you, that more and more you perfuse sorafenib, you see that sorafenib can inhibit sarcomere shortening, and this will take place until the myocyte lose its ability to contract. This data confirms that sorafenib can induce functional cardiotoxicity in adult human primary cardiomyocyte. If we continue to perfuse the same myocyte shown in the previous slide with sorafenib, you will see that sorafenib can induce structural toxicity. If I speed up the video for you, you can see that there is a correlation between decrease in baseline sarcomere length and the ability of sorafenib to induce loss of sarcomeric structure. And this will continue to happen bit by bit until the myocyte, until we see cell death. This data confirm that sorafenib can induce structural cardiotoxicity in adult primary cardiomyocyte. Next, we tested afatinib, a safe TKI. In this experiment, afatinib effect was assessed on 16 different myocytes at the same time. Here you can see that we recorded the baseline contractility, and if I speed up the video for you, you can see that we tested sorafenib at 0.3 micromole, and then at 1 micromole, and finally at 3 micromole, which is equal to 30-fold Cmax. You can see that sorafenib did not have ability to induce either functional or structural toxicity at any of the concentration we tested in this experiment. This slide summarizes for you the data we obtained with the 8 tyrosine kinase inhibitor. Here you can see the toxic TKIs inhibited sarcomere shortening in a concentration-dependent manner, where safe TKIs had no effect on sarcomere shortening. From this typical dose response curve, again, we can derive IC50 values and we can calculate a safety ratio. From this summary table, you can see the toxic TKIs comes with a low value for the ratio. This means that the efficacy and the cardiotoxicity can't be separated, and this is a bad news for cancer drug development. Looking at the 4 safe tyrosine kinase inhibitors, you see that they come with high value of the ratio. This means that the efficacy and any potential cardiotoxicity can be separated, and this indeed a good news for cancer drug development. Our data are in agreement with recent data published in 2018 in Nature Scientific Report. In this paper, sorafenib was shown to induce negative inotropy when tested on human cardiac preparation. And most importantly, the data in this paper suggests that there is no need for any cardiac remodeling to see to demonstrate the ability of sorafenib to induce negative inotropy. As you can see from this slide, our data are in 100% agreement with this published data and confirm the ability of the adult human primary myocyte to predict inotropic potential, negative inotropic potential of cancer agent. If you are next slide shows you the data we obtained with doxorubicin, a toxic anthracycline agent. As you can see from the data presented in this slide, doxorubicin inhibited sarcomere shortening in a concentration-dependent manner, and it has an IC50 of 21 micromole. This IC50 correlates very well with the clinically relevant concentration for doxorubicin to induce cardiotoxicity in a patient. If you are attending the SOT meeting in Baltimore, we will be presenting novel and exciting mechanistic data explaining how cancer agent can modulate contractility in adult human primary cardiomyocyte. The platform presentation will take place on March 14 at 8:30 a.m. In room 314. Next, we wanted to confirm the ability of adult human primary cardiomyocyte to predict positive inotropy. To this aim, we tested 17 different inotropes that they cover 12 different mechanisms of action. This slide shows you an experiment conducted with ISO. You can see after application or upon application of ISO, we can see increase in sarcomere shortening, and this explains the positive inotropic effect of a beta adrenergic stimulation. This slide and the next slide gives you an idea about the quality of the dose response curve we obtained when we tested the 17 positive inotrope. As you can see from this typical dose response curve, you can derive the no effect concentration, we can derive the EC50 value, and also we can derive at which concentration the maximum positive inotropic effect was seen. In conclusion, this data confirms that the adult human primary cardiomyocyte can be used to detect positive inotropic potential of drugs. Also, we noticed that positive inotrope with different mechanisms of action have different mechanisms on the kinetics of the contractility transient. One positive inotrope like this one here speed up the relaxation phase of the contraction. This is how we pick up the positive isotropic effect of this compound. Another positive inotrope increase the contraction time. A third positive inotrope induce after contraction. To get more mechanistic information about mechanism of action, we conducted the cluster analysis. Here in this analysis, we assess the effect of the 17 positive inotrope and also 9 negative inotrope on 12 different parameters that we can derive from the contractility transient. Looking at these 4 heat maps, you can see that the effect of compound on these 12 different parameters is concentration dependent. At low concentration, you can see we can uncover the efficacy potential of inotropes to either increase or decrease contractility. However, at high concentration, you can see that the inotropes can have different effect on different parameters. This is how we can start picking up mechanistic information. To give you an example, we tested 2 sodium channel blockers and we tested a few calcium channel blockers. Both sodium and calcium channel inhibition leads to a decrease in contraction. So for example, if you use the sarcomere shortening parameter, you can't differentiate between a sodium and a calcium channel inhibition. However, using the parameter contraction failure, we can differentiate between a sodium and a calcium channel inhibition. As you can see here, sodium channel inhibition induces contraction failure, and this happens because when you block sodium channel, the myocyte will lose its ability to depolarize, and therefore it can't initiate. An excitation contraction coupling can't be initiated. When it comes to inhibit the calcium channel, you simply reduce the calcium influx to the myocyte. However, the myocyte can continue to be depolarized, and an excitation contraction coupling can be initiated at all time. So this is how we can differentiate a sodium channel inhibition from a calcium channel inhibition. Also, the 17 positive inotrope and the 9 negative inotrope we tested, they either increase or decrease contractility by increasing or decreasing calcium. When we did a PCA analysis, you can see that we have 2 clusters. The blue cluster represent the positive inotropy space, and the red cluster represent the negative inotropy space. When you look at the data, you can see that compounds can affect the clusters in a concentration dependent manner, and only at top test concentration, the blue and red cluster can be separated. This means if a compound increase contractility and the compound sit within the blue cluster, This indicates that this drug increases contractility via increase in intracellular calcium, and this comes with a confidence probability of 75%. To confirm the PCA prediction and to get an idea about the effect of compounds on the kinetics of the calcium transient, we developed a calcium model. In this slide, you see we conducted an experiment with 26 myocyte recording the calcium signal. In this experiment, if I replay the video for you, we recorded the calcium signal under baseline, and then we applied dofetilide at 1 concentration. Then we wanted to confirm the ability of our calcium model to pick up decrease in the calcium signal when we test a negative inotropic compound. Here you can see that we tested Verapamil at multiple concentration, and we see that Verapamil decreased the calcium signal in a concentration-dependent manner. Next, we tested ISO, also to confirm that with a positive inotropic compound, we can pick up an increase in the calcium signal in the adult human primary cardiomyocyte. As you can see here, ISO tested at multiple concentration exerted a concentration-dependent increase in the calcium signal. Based on the validation data I presented, we are proposing that the adult human cardiomyocyte model can be used as a primary screening tool in early drug discovery, because the adult human primary cardiomyocyte allow to evaluate an integrated evaluation of all human cardiac targets, because they have very good predictivity, and also because they can be used in a screening format. A platform is currently being validated. We are proposing first to test that you test all of your drugs on the human primary cardiomyocyte model. If the data indicate that your compound is not associated with a risk, the compound can be moved to next R&D stage. If the data indicates that your compound is associated with a risk, the mechanistic information that we can derive from the myocyte contractility experiment can help to implement follow-up screens, including ion channel screens to dial out the liability. We strongly believe that this model will and this strategy will accelerate preclinical development and will help to efficiently dial out liabilities. To wrap up this webinar, the adult human cardiomyocyte model can provide an integrative assessment with a physiologically functional cells. It has a reliable prediction of inotropic risk. It is ideal for mechanistic investigation, and most importantly, it's predictive of clinical outcomes. Thank you very much for your attention, and I'm looking forward now to answer your questions and comments. Anyone would like to ask a question? Yeah, go ahead, Frederic.
Yes, very nice talk, Najah. I have a question related to the comparison of acute and chronic effect of your inotropic agents. How can you assess in your model some, I will say, subchronic effect, and how do you schedule your different recording time points?
Yeah, basically the data we presented are acute assessment of acute effect of inotropes on contractility. So at the moment, you know, we are working on a model so that will allow us to look for a chronic assessment of drugs. But at the moment, you know, this is 5 to 10 minutes acute assessment.
Acute, okay. And generally, you assess the effect what at 13 minutes after administration?
Yeah, basically we do 2 minutes baseline recording to ensure that you know the contractility and the data we are getting meets the criteria, so to accept the cell to do the experiment, and then we perfuse each concentration for 5 minutes, and we do four ascending concentration basically. So it takes around 20-25 minutes to complete one experiment.
Okay. And how long can you keep a cardiomyocyte? 1 hour, 2 hours, 3 hours?
So this protocol, it's for a routine screen at 1 hertz basic frequency. So within 25 minutes, you can test for concentration and derive a curve. We already we can keep them, going for recording for up to 1 hour. We haven't conducted any specific experiment to see if we can record for 3 hours, for example.
Okay.
But we have checked this when we did force of contraction measurement from trabecular tissue, and this is where also we can go for up 4 or 5 hours recording. But with the myocyte, we tested up to 1 hour only.
Yeah. Okay. Thank you.
Thank you, Frederic. Yeah, hi Russell, please go ahead.
Yeah, hi, Najah. Can you give us an indication of how much inter-assay variability you see from one heart preparation to the other?
Yeah, we looked at this basically at 2 levels. The first one, we looked at variability for intra versus inter-heart variability. For action potential recording, we recorded from ventricular tissue, and we published this in 2016. So basically, we have a variability of 7 to 8%, and 90% of the total variability comes from intra-heart variability, which basically sometimes, the experimented condition, they influence the intra-heart variability. And then we repeated this when we did the myocyte experiment, and when we published the paper in December 2017 in the supplementary file, there is data on intra versus inter-heart variability, and again, we have low variability from donor to donor and within the same prep.
Thank you. And just a quick follow-up. Could you comment on the sex of the individual from which the heart was obtained? Do you see differences between the sexes? And do you see differences between pediatric versus adult heart?
Yeah, at the moment, the hearts we procure comes from both male and female. At the moment, we are using both of them for screening purpose. So if there is a specific need to look for sex difference, we can look at this. When it comes to action potential, we did observe a difference at baseline action potential between male and female, but we haven't really done a very detailed study to confirm sex difference. When it comes to compare adult to pediatric hearts, we can procure hearts below the age of 17, but we haven't really done a lot of work in this space yet.
Interesting. Thanks.
Thank you. And you have to unmute Russell. Yeah, give Frederic again. Yeah, go ahead, Frederic.
No, it's not me. Sorry. I have another question. How many essay do you, I will say, triplicate or more are you doing for each concentration dose response curve?
So for each dose response curve, as I said, we do 4 concentration and a minimum of 5 cells. So based on the data, based on the data we presented today with 58 reference drugs, having n equal 5 is enough, to have a good dose response curve and be able to assess the potential risk with a drug.
So you have a good sensitivity and reproducibility with 5. Okay, understood.
Yes. But of course, if there is, and again, a specific need, we can do on multiple donors.
Okay. Okay. Thank you.
Thank you, Frederic.
It was nice calling.
Thank you. Yeah. Any other question from anyone? Yeah, if you can type a questions if you would like, and then we can answer them straight away now, or we can answer them later on by email after the webinar. Okay. So for any additional question, please email us at info@anabios.com. You can also follow us on LinkedIn, Twitter, and Facebook. And you will receive an email after this webinar in which you can download the video and send us any question you would like us to answer. Thank you very much for your participation, and talk to you soon. Bye-bye.